protein sequence database. Protein sequence database is a biological database that contain information regarding the protein sequences. UniProt KB, Swiss-Prot, PIR, PSD are the example of protein sequence database. Today we will start with UniProt KB. The mission of UniProt is to provide the scientific community with a comprehensive, high quality, and freely accessible resources of protein sequences and functional information. UniProt Consortium. The UniProt is a collaboration between EBI, that is European Bioinformatics Institute, Swiss Institute of Bioinformatics, SIB, and Protein Information Services, PIR. These three together form the UniProt Consortium. This is the home page of UniProt. The UniProt database has the following section: UniProt KB, UniRAF, UniPark, and Proteome. UniProt KB again have the two sections: UniProt KB Swiss-Prot and UniProt KB translated. Is manually annotated and reviewed database, contain the record with the information extracted from the literature and curated computational analysis. Translated EMBL annotated and non-reviewed database contain the record that awaits the full manual annotation. UniRAF is a reference cluster that clustered the set of protein sequences identity, 50% identity, and 90% identity. It is done to speed up the sequence similarity search. The third one is the UniPark, that is a protein sequence of the publicly available protein sequences in the world. Proteome is a set of protein thought to be expressed by the organism for the species with a completely sequenced genome. So these all are the section of UniProt. Okay, UniPark and Proteome. In this figure, I've shown you how the data move in the different section of UniProt database. See, in the UniProt database, 90% of the sequence from the other sequencing database such as EMBL, GeneBank, DD. TrEMBL is a gene prediction database, Vega, RefSeq, and other sequencing resources. From this is coding part get translated into the protein sequence and move to the UniPark is a protein sequence archive which contain the new revised sequences. From the UniPark, the data move to UniRAF, UniProt KB, and UniMass. In the UniMass, all the protein sequence which were extracted from the metagenomic studies were moved to the UniMass. Okay, metagenomic study is the study of genetic material recovered directly from the environmental samples. So the protein sequences which were derived from the metagenomic study, metagenomic projects were moved to UniMass. In the UniRAF, the sequence get clustered at 100%, 90%, and 50% identity. It is done to speed up the sequence similarity search. In the UniProt KB, the sequence first moved to the translated EMBL, which is unreviewed and automatically annotated database. After the manual annotation, the sequence moved from the translated EMBL to the Swiss-Prot, which is a reviewed and manually annotated database. This is how the data move in the UniProt databases. Say the UniProt KB is a protein sequence knowledge base, have the two sections, UniProt KB Swiss-Prot and UniProt KB translated EMBL. UniPark is a protein sequence archive. UniRAF cluster the protein sequences with 100%, 90%, and 50% identity, useful to speed up the sequence similarity search. UniMass contain the protein sequences derived from the metagenomic projects. Metagenomic, what is metagenomic? Metagenomic is the study of genetic material directly from the environmental sample. Here also I have shown you the same thing. See the UniProt database, the UniProt archive, that is UniPark, is produced at EBI, UniRAF is produced at PIR, and UniProt KB Swiss-Prot is produced at SIB, and translated EMBL is produced at EBI. EBI, SIB, and PIR together form the UniProt consortium. Coming to the central piece of the UniProt database, that is the UniProt KB. The UniProt KB is an encyclopedia on protein composed of two sections: UniProt KB translated EMBL, which is unreviewed and automatically annotated database, and the UniProt KB Swiss-Prot, which is reviewed and manually annotated database. The origin of protein sequences in the UniProt KB. The UniProt protein sequences are mainly derived from 85% of the sequence comes from the translation of the coding sequences present in INSDC. INSDC is an international nucleotide sequence database collaboration. Gene Bank, DDBJ, and EMBL nucleotide database together form INSDC. So 85% sequence present in the UniProt come from the translation of the coding sequences present in the nucleotide database. The remaining 15% comes from the assembled and the RAPSAC sequences of PDB structure, and the direct submission or the sequence scanned from the literature. So these are the sources of protein sequences in UniProt KB. Here I have shown how the sequence extract from the nucleotide database to the translated EMBL, and from the translated EMBL to the Swiss-Prot. Okay, EMBL is one of the nucleotide database. From the nucleotide database, the CDS part get translated into the protein sequence and move to the translated EMBL database. Along with the protein sequence, the gene name bibliographic information were also extracted from the EMBL to the translated EMBL. Automated extraction of proteins, that is translated CDS, gene name and references. Okay, this all information were extracted from the EMBL to the translated EMBL, and everything is automated, means computer based, software based. After the manual annotation of the sequence and a biological information, the sequence move from the translated EMBL to the Swiss-Prot. That is why translated EMBL is unreviewed and automatically annotated database, whereas Swiss-Prot is manually annotated database. Okay. Again, I am repeating how the sequence move from EMBL to translated EMBL to the Swiss-Prot. EMBL is a nucleotide database. From the nucleotide name and references were automatically extracted and moved to the translated EMBL. From the translated EMBL, after the manual annotation of the sequences and associated biological information, the sequence moved to the Swiss-Prot. Coming to the UniProt KB translated EMBL, which is unreviewed and automatic annotated database. See, in the translated EMBL, one protein sequence, one species. What is the meaning of this? As I have told you, in the protein database, in the UniProt, 90% of the protein sequence were extracted from the other sequencing database. So what happened? Sometimes entries might be present which corresponds to the different. For example, one report of the protein sequence level. Okay, means the direct submission of the protein sequence. The second report corresponds to the protein sequenced at transcript level, means the one have isolated the mRNA of that particular protein, sequenced the DNA and submitted the cDNA sequence to the gene expression database. Expression database, that cDNA get translated into the protein sequence and submitted to the translated EMBL. So for that particular protein, now second entry is there. The third entry corresponds to the protein sequenced at genomic level, means one have isolated the DNA of that particular protein, have sequenced the DNA and submit that DNA to the nucleotide database. From the nucleotide database, the CDS part get translated into the protein sequence and submitted to the translated EMBL. So what will happen now? For protein, now three entries are present which corresponds to the different database. One is at a direct submission of the protein sequence at a transcript level, third at the genomic level. So what will happen in the translated EMBL? the 100% identical protein sequence extracted from the same species are merged automatically into the single entry. मतलब अगर 100% आइडेंटिकल प्रोटीन सीक्वेंस है, सेम स्पीसीज से अगर आया हुआ है, ओरिजिन अगर सेम है, तो दे आर मर्ज्ड ऑटोमेटिकली इनटू द सिंगल एंट्री. Okay, so one protein sequence, one species. Okay. Next is all the annotation are automated annotation. Automated means computer based. Okay. See the UniProt KB translated EMBL automated annotation at protein sequence and at biological information. Protein sequences, the quality of the protein sequences is depend on the information provided by the submitter of the original database from where that particular sequence has been extracted. Original database में कितनी सही information है उस पे depend करेगा आपका quality of the protein sequence. And the second one is 100% identical sequences. That is same length, same organism are merged automatically. Okay. Second is the biological information. The source of biological information annotation is first provided by the submitter, such as EMBL, PDB. है ना, जो आपका protein sequences, also the gene name, protein name were also extracted from the न्यूक्लियोटाइड डेटाबेस और फ्रॉम द ओरिजिनल डेटाबेस. तो ये आपका बायोलॉजिकल इंफॉर्मेशन का पहला सोर्स क्या हो गया, प्रोवाइडेड बाय द सबमिटर. एंड द सेकेंड सोर्स इज फ्रॉम द ऑटोमेटेड एनोटेशन बिल्ड रूल. देयर आर टू टाइप्स ऑफ बिल्ड रूल, SAAS and uni rule. SAAS is automated generated annotation rule based on decision tree algorithm. Okay, it is algorithm based, formula based. Okay, and the second one is the uni rule, is generated annotation rule generated by the expert biologist. Okay, the protein sequence move through these two rule, SAAS and uni rule, to get the automated annotation record for translation. So these are the two sources for the biological information. First provided by the submitter and second is by the build rule, SAAS and uni rule. Second one is the UniProt KB Swiss-Prot, which is a reviewed and manually annotated database. From the translated EMBL, after the manual annotation of the protein sequences and biological information, the sequence moved to the Swiss-Prot. That is why the Swiss-Prot is reviewed and manually annotated database. Okay. If you see here, I have shown you one protein sequence, one gene, one species. And in the translated EMBL, I have written one protein sequence, one species. And here I have added one gene also. So what happened when the sequence moved to the Swiss-Prot, all the gene product, the alternative product also produced by the single gene, by using the alternative splicing, by using the alternative promoter usage, by alternative initiation sites were also described within the entry. It means that the Swiss-Prot provide a single entry for all the protein product derived from the certain gene from the certain स्पीसीज, यानी एक पर्टिकुलर जीन से जितने भी प्रोटीन सीक्वेंसेस बन रहे हैं उसको भी आप एक ही एंट्री के अंदर हम डालते हैं in order to reduce the redundancy of the database. Okay, so one protein sequence, one gene, one species. Okay, the alternative product, the protein sequences produced by alternative splicing, alternative promoter usage, alternative initiation sites were also described within the entry. And all the annotation are manually reviewed. Okay. So I have written here manual annotation. All the annotations are manually reviewed when the sequence move to the Swiss-Prot. So UniProt KB Swiss-Prot manual annotation. Manual annotation में आएगा आपका protein sequence curation and biological information curation. In the protein sequence curation, in this the experts will merge the available CDS and will correct the sequence annotation and the sequencing report. Okay. In the biological information curation, the experts will do the sequence analysis. There are various analysis program through which the sequence has been moved to predict the sequence function, extract the literatural information, and ortholog data propagation. Okay. Coming to the protein sequence curation, see, one entry, one gene, one species. है ना? Means the in the Swiss-Prot provide a single entry for all the protein product derived from the certain gene from the certain species. And also the alternative sequences were described within the entry. And if there is any conflict, it will be described in the feature table. The displayed protein sequences are canonical, representative, and consensus. At least 20% of the Swiss-Prot entry required a minimal amount of curation effort so as to obtain the correct sequences. When the sequence moved from the translated EMBL to the Swiss-Prot, it required at least 20% of curation. What are the typical problems? The first one is the unsolved conflict, incorrect initiation site, frame shift, wrong gene prediction, and other problems. So all these problems have to be solved before the sequence move from the translated EMBL to the Swiss-Prot. After solving this problem, the sequence move to the Swiss-Prot. The next one is the biological data curation. In the biological data curation, the experts will extract the literatural information and go for the protein sequence analysis. There are various analysis programs to predict the protein function. There will be mandatory usage of controlled vocabulary. The Swiss-Prot gather data from multiple sources such as publication, prediction program, contact with experts, other database, and nomenclature committee. This all has been done for the manual annotation of the biological information. Move from the translated EMBL to the Swiss-Prot. See, the UniProt KB entry contains the protein name, gene name, also known as all the synonyms of the gene name. Okay. Like protein functions, subunit structure, cellular location, tissue specificity, domain, post translation modification. मतलब in the UniProt KB, you will find everything what is known about that protein. The sequence annotation will contain the DNA binding site, zinc finger site, region, then modified residues, cross links databases, which enable the researchers to obtain the summary of what is known about that protein. प्रोटीन की जितनी भी इंफॉर्मेशन प्रेजेंट है वो सारी की सारी आपको यूनिप्रोट एंट्री में मिलेगी. The next is the protein existence tag. The protein existence tag indicate what is the evidence for the existence of the given protein. When the sequence move from the translated EMBL to the Swiss-Prot, it is very, very important to provide the protein existence. We have to give the evidence for the existence of the given protein. So there are different qualifiers for the protein existence tag. First is the protein level, means the protein sequence of that particular protein is present in the database, means a direct submission of the sequence at the protein level. Okay, so 18% of the protein in the Swiss-Prot have the existence at protein level. Second is evidence at transcript level, means the cDNA sequence of that particular protein is present in the database. 19% of the entry in the Swiss-Prot have the evidence at the transcript level. 58% at homology, means the homologous sequence of that particular protein is present in the database. 5% is predicted, and if it is uncertain, then the sequence remain in the translated EMBL. It will not move to the Swiss-Prot. In this figure, I have shown you how the sequence enter into the translated EMBL and from the translated EMBL to the Swiss-Prot. So when the move to the translated EMBL, the very first step is the automated annotation. So for the automated annotation, the first step is to identify the template. Identify template means the identification and selection of the template set of entry sharing some common annotation. The second step is to build rule. Okay, the build rule is done to identify the reliable common annotation. There are two ways to build rule, SaaS and UniRule. SAS is an automated rule generation by C4.5 decision tree algorithm. It is algorithm based. And second one is the UniRule, the rule created manually by the expert biologist. So the sequence move from these two rules to build up the automated annotated protein record. Okay. Apply the suitable rule to the UniProt KB translated EMBL. After applying this rule, we will get the automated annotation protein record and the sequence move to the UniProt KB translated EMBL. So for the sequence to move from translated EMBL UniProt, the step is the manual annotation. After the manual curation of the sequence, the, the sequence move from the translated EMBL to the Swiss-Prot. The first step of manual curation is the sequence curation. Where the same gene are merged and discrepancies are curated. The second step is analysis, in which sequence analysis program are run to predict the sequence feature. The third step is the literature curation, where the full text of the paper is read and information is extracted. The next step is the family-based curation, where the homologs are identified and curated. Then evidence attribution, all information is attributed to the original source. See the protein existence tag, evidence, we will find the evidence. So all the information is attributed to the original source. Then quality assurance and integration step, where the entries undergoes the quality assurance before integration into the UniProt KB Swiss-Prot. After moving all the steps, the sequence move to the UniProt KB Swiss-Prot. Again, the rule, the build rule is applied to the sequence, and the sequence will be in UniProt KB Swiss-Prot. So this is how the sequence entered into the translated EMBL to the Swiss-Prot. This is the format of entry in the UniProt KB. See, protein name, gene name, organism, status, unreviewed means the sequence remain in the translated EMBL. Annotation record means the protein existence tag. See the protein inferred from homology. Then molecular function, lyase activity, protein name, gene name, organism, taxonomic identifier, taxonomic lineage means classification, structure. If the structure of that particular protein is present, and here is the link for the 3D structural database, and the family and the domain database, Gene 3D, Interpro, Panther, PFARM, Superfarm. Here all the links are present where you can go to get the information regarding this. Then the sequence, UniPark. This is the UniPark ID. Here is the protein sequence, protein length, mass, last modified, and checksum. Then the experimental information, then the sequence database. If you want to see the nucleotide sequence of this protein, you can visit here. You can see the sequence in various format like EMBL, GeneBank, and DDPJ. Then similar protein, 90% identity and 50% identity, that is UniRAF. Okay. Then the cross-referencing database. Here are the link for the cross-referencing database of containing the information regarding this protein. These are sequence database, then 3D structural database, family, and the domain database. Here all the link are present. Then entry information, entry name, accession number, entry history, integrated into the UniProt KB translated EMBL in October 25, last sequence update, and last modified date. Okay. This is the version 8 of the entry, means the entry has been modified 8 times, and the version 1 of the sequence. You can see the complete history here. Okay. You can see all the version of the entry, how this entry has been modified in this database. The entry status, unreviewed, means remain in the translated EMBL. So this is all about UniProt